Hello students, today we will study about translation or translational machinery. So, before I will start, I will ask you some questions which is very important. You should have the basic knowledge and also the knowledge of the previous topic like transcription. Okay. So, I hope you know very well about the transcription, because translation takes place after transcription, and you know very well that there is a cell, and in the cell there is a nucleus, and in the nucleus there is nucleolus, and the chromosomes are there which contains DNA, deoxyribonucleic acid, which has got triplet codon, and that is the acting as gene, or which is containing the genetic information. Okay. Now, when we are eating something, we are digesting that food. Okay. Now, when you are eating, there are three principal types of food: carbohydrate, protein, and fat. Okay. Three chief principal main types of foods are there. Now, when we say these three types of food, these three types of foods are digested within our body after ingesting. The carbohydrate digestion starts in the buccal cavity or mouth. Now, you must be surprised why I am talking about this digestion and all. Now, this is how I am going to correlate to make the concept very clear, and you listen very carefully. Understood? Okay. If you have any question, yes. Okay. Now, when you are eating something, digestion starts, the carbohydrate digestion starts in the buccal cavity or mouth with the help of the salivary amylase or ptyalin secreted in the by the salivary gland on either side of the jaw. Okay. Now, protein digestion starts in the stomach. That is the region we have hydrochloric acid in our body, in the stomach, because acidic medium is required. Now, when the protein digestion starts in the stomach and is completed, the digestion is completed in the intestine, it is absorbed in the small intestine through villi, small finger like projections that is present in the small intestine, right. Okay. So, protein is digestion. We are concerned here with the protein. Why? Because we are talking about the formation of protein. That is why I started with this, because we are taking food that contains protein, we are breaking down into amino acid. Again, with the help of translation, we are joining those amino acids to form protein. Then why are we breaking down? Now, I will clear this concept. Just listen very carefully. Okay. Now, when we are eating protein, okay, we are digesting into, we are breaking down into amino acid, and amino acid is the end product, and which is absorbed through the small intestine. It is absorbed in the blood, and blood is transporting to different parts of the body. Okay. Now, if you see, I am drawing a diagram. This is the nose and this is the mouth. Okay, and here we have got the buccal cavity, and this is the esophagus, and this is the stomach, and then you think. Now, once this digestion starts in the stomach, okay, it is partly digested, it is acted upon by the digestive juices, and as it moves on to the duodenum and other parts of the small intestine like ileum, then digestion is complete, protein digestion is complete. Okay. Now, once this digestion is completed and the product is formed into amino acid, these amino acids are absorbed. I am repeating, okay, again and again to make you very clear. Once these amino acids are absorbed into the blood and reach to the different part of the body, okay, it goes into the cell. Here is the main point you should remember. Okay, because once this amino acid goes to different cells, okay, it forms the pool in the cytoplasm of the cell, right, from where the transfer RNA brings the amino acid for the formation of polypeptide chain. Okay. When translation occurs, then what is the use and what is the need of breaking down protein into amino acid and again joining back. Here is a complete answer. We are taking protein with the amino acid sequence which is completely different from the sequence that we need to prepare. Okay, we need to prepare different types of protein in our body. For example, we need to prepare a keratin, a protein called keratin, okay, or some other protein, but the protein that we are taking is different. So, we are breaking, but changing the sequence of the amino acid, protein is changing, right. So, we are taking protein, okay, we are breaking down into amino acids, again we are joining in the process of translation the genetic information that has been carried from the DNA to the ribosomes with the help of mRNA, that is decoded and it is read. It is decoded, and according to the sequence that is there in the mRNA, the amino acids are joined and forming the protein which is required by the body. So, it means that we are preparing a different protein, right. We are preparing a different protein, we are preparing a different polypeptide chain. Polypeptide chain means a chain that is continuous with amino acid sequence and joined by peptide bond, and again when polypeptide bond joins together side by side and forming a different complex structure, it is protein in the complex. Okay. So, now we are going into the main topic, translation. So, you understood? Is there any doubt? Please ask me. We are taking in protein, we are breaking down into amino acid, it is absorbed into the blood, it goes to the different parts of the body, it goes into the cell, and then when translation occurs, these amino acids are brought to the ribosome, which is called factories or the kitchen or the place where this protein synthesis or polypeptide chain synthesis takes place, brought from the cytoplasm to the ribosome, where the different sequence it joins with the help of peptide bond and form a new protein which is different from that we have taken in. Okay. Now, the translation means it is decoding. What is doing? There is cell, as I told you here, this is the nucleus and then in the nucleus and the nucleolus, and this is the cytoplasm. When transcription takes place, once that mRNA is formed after transcription, it is coming out of the nucleus and goes into the cytoplasm, and when it reaches to the ribosome, okay, transfer RNA brings the amino acid and it has anticodon, it identifies which amino acid needs to be joined to another amino acid. Okay. So, this way a chain is formed. Understood? Okay. Now, here translation means decoding the information that has been brought from the DNA, genetic information. Okay. So, that genetic information is decoded into protein, functional product. Okay. The DNA has got the information what kind of protein needs to be prepared, in what situation, in which situation, and depending on the situation, depending on the requirement of the body. Okay. So, that is information is given by the DNA and it is transcribed into mRNA. mRNA brings the information to the ribosome, and then the tRNA identifies that information, decoded, and polypeptide chain is synthesized. This is the overview I told you in very short. Okay. Now, mRNA is decoded to build a protein. Yes, I told you. Now, here in this diagram you see the DNA replication. DNA replication is different and transcription is different. DNA replication means forming a new DNA from DNA to DNA, and this transcription means forming mRNA, single stranded template. This is the single stranded template that goes out of the nucleus into the cytoplasm after transcription, right. Okay. Now, once this transcription takes place and mRNA goes out of the nucleus into the cytoplasm, what will happen? The protein is formed. So, this is the overview given here in the diagram. Okay. Now, next we have, now what are the different structure that is involved in the translation. Okay. This topic is very interesting. Ballet is very wonderful subject. I love my subject, and if you study nicely, if try to understand, you will fall in love with this subject, and also you will enjoy studying this subject. Okay, when you are studying and understanding at the same time, that means you will start enjoying the topic and the subject. Okay, now what are the structure? That means these are the ribosomes, as I told you earlier. Also, it is the factories, also regarded as the factories of the protein. mRNA, that is the single stranded template, and it brings the information from the DNA to the cytoplasm to the ribosome, and transfer RNA, which is responsible for carrying the amino acid to the ribosome, and amino acids, that is the monomeric unit that needs to be joined to form polypeptide chain and subsequently protein. Okay, right? And here the last one, this also plays a very important role, because this is an enzyme that plays the charging, because without charging of the amino acid, it will not come to the tRNA. Okay, this amino acid tRNA synthetase is an enzyme that helps in the charging of amino acid that helps the binding of the tRNA and amino acid, so that tRNA will bring the amino acid to the ribosome. Correct? Now, ribosome has got structure like this. They have got one larger subunit and one is the smaller subunit. Now, when we say smaller subunit and larger subunit, it is present in both prokaryote and eukaryote. In prokaryotic cell, it is a little different, and eukaryotic cell, it is also little different. Now, when we say this eukaryotic and prokaryotic, when you are talking about the eukaryotic prokaryotic, transcription and translation, all the process takes place in both the type of cells. Okay, but the ribosomal structure is a little different. In case of eukaryotic, it is a little larger, and depending on the sweat bug unit, it is little different. So that is not we are concerned with. Here we are talking about the larger subunit and smaller subunit, which comes together at the time of synthesis of protein, and where the magnesium ion is required. It associates during the synthesis of protein or polypeptide chain. Okay, so this is the main, one of the very important structure for the synthesis of protein during translation. Translation is taking place. Okay, translation means translating the code into the functional product. Okay, now structure of tRNA is also shown here, and we will see in a little while in detail. Okay, now the function of, as I already I have told you in brief. Now again repeating the same thing here, because it is these are the slides, so to make you very clear. If you have doubt, please ask me. Okay, there should not be any doubt at the end of this lesson. Okay, because you are able to because in the NEET examination, the concept based question are asked. Okay, and it's quite interesting. Okay, if you study very nicely, okay, and understanding the topic very nicely, that's why I enjoy very much, because and those are studying this, and if you have been understood, you will enjoy while answering the questions that is given in the NEET examination. Okay, now first we have to go to the site of ribosome are the site of protein synthesis. That means they are responsible for they are the main structure where the protein synthesis takes place, the place where protein synthesis or polypeptide chain, rather we should say polypeptide chain synthesis takes place. Okay, now mRNA is the template, single stranded template, that is carrying the information from DNA, that so that what kind of protein, what kind of polypeptide chain will be synthesized, in what sequence that amino acids monomeric unit will join, in what sequence, which will be after which one, that information has been kept, because the sequence will change the function of the protein. Okay, depending on the sequence of the amino acid, there are different proteins and the function is different, right? Understood? Okay. Now here tRNA is, as I told you, charging is required, and here the tRNA brings after binding with this, after charging binding with the amino acid, it is bringing the amino acid to the ribosome for the synthesis of polypeptide chain, and amino acyl tRNA synthetase is an enzyme which is responsible for charging of amino acid. Okay. Now look at this. Ribosomes are the factories, as I told you, because they have got larger subunit and smaller subunit. They are joined together. They are forming a group in between, okay, where the actual synthesis takes place. And there are three different sites. Here is the A site. I have missed this one. This is the A site where the amino acid is entering through which, okay, and there is a P site, peptidyl site. We will discuss in detail in little later. And the E site, exit site, where the synthesized polypeptide chain is moving away from the ribosome. Okay, so two subunits are there. That is the larger and smaller subunit. Magnesium ion is required, as I told you. This is very important. Now you have to note down this one. Question for NEET. This is asked in the NEET examination. Which ion is involved in the synthesis of this, what we call that, which ion is required or involved in the association of the ribosomal units, the larger subunit and smaller subunit. Okay, and once the synthesis is complete, these two subunits dissociate from each other, and they then again moves into the cytoplasm. Okay, now next. Now ribosome, as I told you, larger subunit, smaller subunit. It has groove when they are associating, and it has larger subunit. It has got groove, as you can see in the picture. Okay, so that newly formed polypeptide is protected and also protected by the cellular enzymes. Okay, so there is the groove, so that there will be protection from the cellular enzymes that is not going to affect the newly synthesized polypeptide chain. And a smaller subunit, it fits like a cap, okay, and it has also got some tunnel like space where the mRNA can moves, so that tRNA which is coming, and they are reading the mRNA and they are bringing to amino acid for the formation of polypeptide chain. Okay. Now when this ribosomes forming, and they are one by one, they are forming a chain like a structure, they are grouping. That is called polyribosomes or polysomes. Now parts of ribosomes I have already discussed. I am now 2-3 slides I have given, okay, so that you will have more clear, you will have a clear understanding, and you should not have any doubt in your mind. Okay. Okay. Now when we see this, there is a E site, P site, and A site again here. Maybe I have. Okay, this is the A site where the amino acid along with the charged amino acid along with the tRNA enters to the ribosome. Okay, exit channel for the passage of newly synthesized polypeptide. Okay, and exit site means E site, and these three reactive sites, P, this is called also D site, P site, peptidyl site is also called D site. Okay, A site, they are the reactive sites. That means where some kind of process takes place. Entry of charged amino acid along with the tRNA takes place through A site, okay, and exit takes place through E site. Okay, and the actual recognition of the mRNA for the synthesis of polypeptide chain, that takes place at the P site, that is right on the, which is there right on the mRNA. Okay, now we see that there are P site, as I told you, peptidyl donor site is jointly contributed by two ribosomal units. That means smaller one and the larger one forming this site together. A site where the charged amino acid and the transfer is brought to the ribosome, and exit site, that is from where the synthesized polypeptide chain moves away slowly. Okay, there is a tunnel. Now this is the exit site. Okay, and this is the methionine. It means this is the methionine. The amino acid methionine has been brought. This is the AUG, and conversely UAC is anticodon. Okay, AUG is the start codon. So start while this charged amino acid is coming. Okay, so it is searching for the start codon to initiate the translation. Unless and until it won't get any AUG codon, it will not start. As soon as it gets AUG, the translation is started. This is called start codon, where when the translation is start. Okay. Now tRNA, like I told you before also, transfer rRNA, pick up the particular amino acid, and there is a process called charging, where these amino acids are bound to the tRNA. Each tRNA molecule carry only one kind of amino acid, and as determined by the anticodon. So what anticodon the tRNA have, accordingly they are carrying the amino acid. Okay. So that is the reason they can identify the mRNA sequence, and consequently, according to the information or the code that is present in the mRNA, amino acid chain is formed. That means polypeptide chain is formed. Now each tRNA has an area for coming in contact with ribosomes and the enzymes amino acid tRNA synthetase. I told this before also. They are very simple, isn't it? tRNA has an area to for coming to the ribosome, and also for the amino acid RNA synthetase. That means I will draw a diagram. I will show you. For example, if this is the structure like this, this is the structure. So there will be one site for the amino acid to join, and one side for enzyme to react. Enzyme displays a role for the charging of the amino acid, so that amino acid will join to the tRNA, which can be carried to the ribosome for the synthesis of polypeptide chain. Very simple, right? Okay. Now amino acid tRNA synthetase, it is the enzyme that helps in coming to the amino acid, as I have already told, that helps in combining amino acid to its particular tRNA. So that means one particular amino acid will combine with particular type of tRNA, because it has got anticodon, it has got some code. Accordingly, the amino acid will join or will be charged, and these charged amino acid along with the tRNA goes to the ribosome and look at the search in the mRNA, and if it matches with the anticodon and codon, then it will form the polypeptide chain, right? Okay. It is specific for each amino acid, as I told you. Okay. Now it is also called AA activating enzymes, amino acid activating enzyme. Instead of telling, okay, tRNA synthetase, we can call as amino acid activating enzyme. Okay. Now in the mechanism of translation, there are different processes. The main process are which is mainly observed, which is the main process, that is initiation, elongation and termination. Modification is something that is the modification that takes place after synthesis of polypeptide chain. Okay, modification that can take place. For example, there is a structure called protein structure called primary structure, secondary, quaternary, okay, tertiary, quaternary. Like hemoglobin is a quaternary, complex, very complex molecule. Okay, alpha helix in our hairs, keratin. Okay, that is the beta structure, secondary. So this modification, once the polypeptide chain is formed, the chain, the binding or joining of the polypeptide chain or other conformational change in the shape, that is the modification. But what about the top two, activation of amino acid and charging? That's what I was talking since then. You know, like charging, amino acid is activated, it is charged, so that it can join with the tRNA, and tRNA can bring it to the or take it to the ribosome. Okay. So these two, the first one and the second one, it is the needed for tRNA to bring the amino acid to the ribosome, and after that initiation, that means as I told you, AUG, the first start codon, recognizing the start codon AUG, and then the translation or continuous it is initiated, and that continuous translation takes place until it is found a stop codon. Like a start codon, there is stop codon also. Okay. So once it gets the stop codon, then translation stops. It is called termination. And once it is started, once the process is started, initiated, it will form a chain until a stop codon is found. So that growing, the joining one after another, the amino acid, that is called your, what do you call that, elongation. It is elongating because one after another the amino acid is being added and peptide bonds are being formed in between the amino acids. So, that is the called elongation. Okay, any doubt? Okay. Now, activation of amino acids. Now, here how this activation takes place, it is carried out by the activating enzymes I told you earlier also, and known as amino acyl tRNA synthetase. And in the presence of ATP, this is the energy requiring process. Whenever there is energy required, this means ATP, in the presence of ATP molecule. Okay. So, an amino acid combines with a specific amino acyl tRNA synthetase and magnesium ion is required. Okay. And that means this magnesium ion is playing a very important role. Now, it produces amino acyl adenylate complex. Now, how this is? So, this is the amino acid plus ATP plus amino acyl synthetase enzyme. This is the enzyme, and this is the amino acyl adenylate complex, and okay, and inorganic pyrophosphate is formed. Okay, this reaction takes place. Okay, this biochemical reaction takes place for the activating amino acid. Okay, activating enzyme. Okay, first enzyme is activated. Okay, now charging. Now, once the enzyme is activated, okay, here we saw that enzyme is activated. Now, we are saying the amino charging of the amino charging or amino, this charging of amino acid along with to be combined with the tRNA, it is also called amino acylation. Okay, so amino acylation of tRNA. Now, the complex reacts with tRNA specific for the amino acid. Okay, that means for the specific amino acid there will be a specific tRNA and the complex is formed. Hydrolysis of pyrophosphate provides energy for driving initial reaction. Yes, hydrolysis of pyrophosphate, which I will. Okay, now enzymes and AMP are released here. AMP and enzymes are removed, and this amino acyl adenylate enzyme complex, this is the complex plus tRNA. Now, it is activated. This is charged. Now, this is charged. So, in the previous slide we saw that it was activated, enzyme was activated. Now, here the requirement before the translation starts, before the initiation starts, it must be charged. No, so charging has been done now. How? Now this enzyme is released and it has been this complex is formed. It's coupled with the amino acid is coupled or forms a complex with tRNA. Okay, so this is the product. This is the complex. This complex, for the formation of this complex, these chemical reactions needed to be takes place for the formation of this product, because this amino acyl tRNA, charged amino acid, this goes to the ribosome. So, formation of for the formation of this particular product, all this reaction took place. Okay. Now, after this, the next process, that is the initiation. Now, once the charging has taken place, now it is ready to go to the ribosome. Okay. Now, what happened in initiation? It will identify. For that reason, there are some initiation factor, but you should not remember all this IF3, IF2, IF1, but you should remember that there are some initiation factor in the cell for the initiation of translation. For the prokaryotic and eukaryotic cell, for both these two types of cells, the different types of initiation factors are there. So, for the prokaryotic, it is IF3, IF2, IF1, and eukaryotic EIF2, EIF3, EIF1, EIF4A, etc. Okay, likewise. Now, this diagram, this picture is showing that there is a 50s and the 30s. This is the Swedberg, according to the Swedberg-Vinney, the 50s, the larger unit and the smaller unit. Okay, and the green one is showing the mRNA template. Okay. Now, when mRNA attaches itself to the smaller unit of ribosome in its cap region, and the larger subunit comes above it. Okay. Now, cap has a nucleotide complementary to the nucleotide present at the 3' end of rRNA. There is ribosomal RNA. ribosomal RNA in the smaller subunit, and that recognize when RNA template is coming. So, it is recognizing, and then mRNA is just moving on the smaller subunit. Okay. Now, once the attachment is such that initiation codon of mRNA comes to lie at the P site. So, what happens? The attachment of the mRNA to the smaller subunit is such that start codon AUG will just lie to the P site, so that initiation will takes place. Okay. Now, when initiation takes place, amino acid tRNA complex specific for the initiation codon reaches the P site. So, when the amino acid which is charged, coupled or has formed a complex with tRNA come to the P site, for AUG, the first amino acid for initiation codon AUG, the amino acid which is corresponding to AUG is the methionine. That is why it is showing here methionine. Okay. So, from the first amino acid that comes in the initiation process, that is the first one that is coming to form a polypeptide chain is the methionine. Okay. Now, anticodon establish the temporary hydrogen bonding with the, yes, this is needed because still the formation of chain is going on. Now, here I told you before also magnesium ion is required of the larger subunit of ribosomes, now combines with the mRNA and tRNA methionine complex. Please pay attention here, listen very carefully. It is saying that now combines with mRNA tRNA methionine complex. So, mRNA is there, tRNA has brought the amino acid. Which amino acid? Methionine. So, methionine mRNA tRNA methionine complex. So, magnesium ion is now combines with those complex and form an intact structure with the ribosome Okay. Intact ribosome encloses mRNA tRNA complex present in the P site, but keeps A site exposed. Why? Because the formed polypeptide chain has to exit from that site. Okay. So, I hope this is clear, very clear. Okay. If you have doubt, you will ask me. Now, elongation is the another next step. Okay. Where that means this is very simple. Once the initiation has been the process is initiated, the process is started. Now, another amino acid keeps on coming and starts joining with the help of peptide bond. So, here an amino acid tRNA complex reaches the A site. As I told you, always A site is open also for bringing the charged amino acid. So, charged amino acid comes through the A site. Okay. Now, a peptide bond is formed between the carboxyl of the amino acid attached to the tRNA at P site and amino group of the amino acid attached to the tRNA site. You can see in the picture very clearly that A site is the region where the charged amino acid is coming. Here methionine has come. Now, phenylalanine has come. Now, these two are forming a peptide bond in between, and now lysine is coming, and this will again join with the peptide bond, and this way it will keep on elongating into a chain. That is why it is called elongation. Okay. Now, this formation of peptide bond takes place between carboxyl group and amino group. Okay. Now, the connection between mRNA, the connection between tRNA and amino acid at P site breaks. Okay. So that they can move away from the E site. Okay. The free tRNA of the P site shifts to the E site or exit site, and from there to the outside of the ribosomes with the help of G factor. Yes, this is another point, G factor. Okay. Here this is a chemical reaction again. Okay. And G factor is needed for the exit. Now, A site carries peptidyl tRNA complex. This is in more detail, okay, for the NEET example. When we study about the translation for the CBSE or ISE examination, this much in detail is not needed. There is in detail, but in NEET examination we have to be more in depth, okay, and we have to very clear about the concept, and we have to understand it very clearly, so that any question which is asked about this topic we should be able to answer. Okay, because time is also a factor. If you are not able to, if you are scoring 600 plus, I think in West Bengal you can you should get a medical college easily. Okay, so my students have scored, many students they have 600 plus, they have got. So keep in your mind, biology, because it is 90 marks, it double the other subjects. So you know the number of questions which is there for the physics and the chemistry is double. So you should pay more attention to the biology. Okay. Okay, now coming to the again to the point. Okay, now here A site carries the peptidyl tRNA complex. I have already told. A site is the site which is the where the entry of the charged amino acid takes place. Okay, carries A site carries the peptidyl tRNA complex, which is coming from the cytoplasm. Now elongation, when the polypeptide chain is being formed, the chain is elongating. After formation of peptide linkage and slipping of the free tRNA of the P site, the ribosomes or mRNA rotates slightly. Yes, and the process is called translocation. It is rotating slightly, and the process is called translocation. Okay, the ribosomes or mRNA rotates slightly. Now this is translocation. It requires a factor called translocase and energy. Translocase is another enzyme that is responsible for rotation of the ribosome or mRNA. The process of bond formation and translocation is repeated again and again, because the new amino acid is coming and joining with the help of peptide bonds. So this process is repeated. One by one, all the codons of mRNA are exposed at the A site, and the new amino acid will come and decoded through incorporation of amino acid in the peptide chain, and that peptide chain keeps on elongating. Okay. Now termination. Once it, as I have already told you, once it finds the stop codon, or is called nonsense codon also, then it stops. So there are three nonsense or stop codon: UAA, UAG, and UGA. Okay. Now termination. Once it stop codon has been found, now it will, the translation process which has begun with the initiation, now elongation has taken place, it has found the stop codon or nonsense codon, now it will stop, termination will take place. The nonsense codon or stop codon are not recognized by any of the tRNAs. That is the reason the no amino acid will join now, because the codon and anticodon will not match, they will not recognize. Okay, which amino acid to join, it will not recognize, because it is nonsense codon. Okay, that is why it is called also nonsense. It is stop codon because process is stopped. It is also called nonsense codon. They will not recognize which amino acid to join. Okay, so here the process stops. It is terminated. Therefore, no amino acid acyl reaches the A site, and the P site RNA is hydrolyzed, and the complete polypeptide is released. Then the chain which is formed is fully released from the ribosome. The ribosomes moves over the nonsense codon and slips of the mRNA chain, and the subunits, the two subunits also dissociates in the presence of dissociation factor. There is a dissociation factor. See, in the biology, every day, in the different parts of the world, research is going on. Okay, so whenever there is research result are published, the books of the biology and the topics are updated with the new information. There are dissociation factor. Still some research are going on, not much in detail now. Even now also, it cannot be, it's not at the level of the this class 12 or NEET, but every time, every now and then, the research is going on. So, but there are some dissociation factors. This has been understood that there is some dissociation factor which causes the dissociation of two ribosomal subunit, larger and smaller subunit, after completion of polypeptide chain. Got it? Okay. Okay. Now, thank you for listening. I hope you enjoyed this lesson, and if you have any question, you can ask me. Okay, and please feel free to ask any question which with regard to this topic, or any confusion that you have in your mind. Okay, because you must remember, as I have told you before also, isn't Is it Rajiv? Huh? Yes. Okay. So you know very well. Okay, I have told you before also that teaching is a two-way process, where teacher must know about the understanding or level of the student, and also the feedback is very important, right? So the teacher can accordingly teach the students, right? So if you have any question, you can ask me now, please. Okay, no questions. Okay, 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 okay.